So we're talking about the immune system, and on Tuesday, we talked about the first part of the immune system, which we call the innate defenses. And the innate defenses, remember, these are what you are born with. These are not specific. And so that's going to include our first line of defense, which is basically your skin and your body's membranes. And so we talked a lot about skin, what makes it a fabulous physical barrier, what makes it a great first line of defense. We talked about some of the secretions that come from your skin, like the acid mantle that helps to kill off bacteria and foreign organisms from even getting into your body. And then we also talked about, okay, well, your skin's not perfect and it can break, it can get cut. So we also touched on our second line of defense, which includes a whole host of things. Anything from natural killer cells, phagocytic cells, macrophages. We talked about things like the inflammatory response. We talked about pyrogens, which help you run a fever. We also talked about things like interferons and other chemicals like complement proteins. And all of these things that I've listed here under B, these are all part of our second line of defense. It would be what is activated if something is able to make it through your skin. But these are also part of our innate defenses. You are born with these. These are not specific. It's just if something makes it through your skin, these are there to help fight it off. Now, at the same time that the second line of defense is activated, we're also going to be activating our third line of defense, which is part of our adaptive immune system. And our third line of defense, which I have a list under C here, this is going to include things like our B cells and our T cells. So we're going to spend today talking about B cells and T cells. And again, remember the huge difference between our adaptive defenses and our innate defenses is specificity. Adaptive defenses are specific. B cells are going to release very particular antibodies for very particular foreign antigens. Okay, they are very specific. Okay, T cells are going to target very specific foreign organisms that get in your body. And so that's not something that we see up here with our first and second line of defense. This is not specific. This is just keep everything out that doesn't belong. Okay. All right, so that was just a quick recap. Let's this is where we left off in the lecture. So again, we're kind of moving down into that third line of defense, our adaptive defenses. And again, what makes our adaptive defenses so unique is that they are specific. They are targeting very particular foreign antigens or even abnormal body cells. So remember, all of the cells in our body have a very particular antigen on the surface. They have very particular sugar coatings. If a body cell inside your body changes, maybe it becomes cancerous, it actually usually will also change that sugar coating on the surface. The sugar coating will no longer resemble your self sugar coating. It'll start to look different. So your body will also recognize that as foreign. Now, for your adaptive defenses to work, they have to be primed. So they have to see something that doesn't belong and fight it off. And the priming takes time. And so this is why this is considered our third line of defense. It is the slowest to respond because it has to be primed. Okay, so there are three aspects to our adaptive immune system. One is, and I know I've mentioned this multiple times, it is specific. It is targeting very particular foreign antigens. It is systemic. It is not restricted to the initial site of infection. Your B cells and T cells, your antibodies, they move throughout your entire body. So it's systemic. And then lastly is memory. Once your adaptive defenses have seen a very particular foreign antigen, they create memory to it. So the next time you're infected with that exact same foreign antigen, you fight it off so fast that you don't even have any symptoms. You don't even know you were sick. Okay? And so when we talk about our adaptive defenses, I keep telling you these are our B cells and our T cells. So there's sort of two separate but overlapping arms, B cells and T cells. Your B cells provide you with something called humoral immunity. Your body's humors are essentially the liquid, the fluid in your body. So all the fluid in your body, your blood, your lymphatic fluid, all the fluid is considered your humors. So what we say is that your adaptive defenses, your B cells provide you with this humoral immunity. B cells create antibodies, and those antibodies circulate throughout your body's humors. So this is how they are providing you with humoral immunity. They move through your body's fluid. Your T cells are providing you with cellular immunity, because T cells are going to fight off infected body cells. That is their job. They fight off infected cells in your body. So these are cell mediated, whereas B cells make antibodies, T cells fight off infected cells. So we're going to walk through the humoral immunity, and we'll talk a little bit about our B cells, and then we will spend way more time talking about these as well. So this is just sort of a 10,000 foot overview. So for humoral immunity, again, these are our B cells that are providing this. These create antibodies, and again, why we call it humoral immunity is that antibodies circulate through your body's fluids. And the role of an antibody is a protein, basically, that's now moving through your blood, it's moving through your lymph. And the job of an antibody is that it is going to temporarily bind to something that doesn't belong. So it'll bind to and inactivate any free-floating bacteria, any free-floating bacterial toxins, or any free-floating viruses. Notice that every time I said free-floating, that means if you have a bacteria floating in your bloodstream, if you have a virus floating in your bloodstream or your lymphatic fluid, that's what antibodies will target. Anything that's free-floating that doesn't belong. That antibody is going to see it, and it will latch on temporarily, it will inactivate it and flag it for destruction. And destruction, we'll talk more about this a little bit later, but when an antibody flags it for destruction, what that really means is it's sort of like the antibody latches onto it, and it's just like a flag sticking out of that bacteria, so that you have phagocytic cells that know, oh, that doesn't belong, that shouldn't be here. And so that's really what they're doing. Antibodies just sort of latch on and flag that free-floating foreign organism for destruction. And it's destruction by a phagocytic cell or some other immune system cell in the body. And so what we say is that humoral immunity, these antibodies, they have extracellular targets. They are going after free-floating viruses, free-floating bacteria, bacterial toxins, things that don't belong that are floating in your body's humors. Your T cells provide you with cellular immunity. So these are very different. T cells are going to target infected body cells. So these are not going after free-floating bacteria or free-floating viruses. T cells are going after a cell in your body that's already been infected by a virus. So T cells are going to kill infected cells. And then they can also activate other lymphocytes and macrophages to really help to ramp up the immune response. So again, the target is going to be A cell in your body that's been infected by a virus. A cell in your body that's been infected by a parasite. A cell in your body that doesn't look right anymore. Maybe it has genetically mutated and it has become cancerous. Or cells, body cells that are in your body that all don't look right because they don't belong in your body. So like grafts, foreign tissues, transplants. T cells are going after body cells that don't belong or don't look right. Okay, so they're not going after free-floating bacteria. They're going after a body cell that's been infected by a bacteria or infected by a virus or has gone awry and become cancerous. Okay, do you see the difference between what B cells are doing and what T cells are doing? B cells make antibodies that float through your fluid and find anything that's floating around that doesn't belong. T cells go after infected body cells. So this is why we say they provide us with cellular immunity. Cellular immunity has cellular targets. Now I've used this term a lot, antigen. We also used this a lot back when we talked about blood and blood typing. Antigens is it literally stands for antibody generating substance, antigen. So we have foreign antigens. These are things that get in your body that don't belong, that cause us to make antibodies to them. And then we also have self-antigens, right? Self-antigens are on the surface of our cells and they belong in our body. So when we talk about antigens in this chapter, we are always talking about foreign antigens, the ones that don't belong, that cause us to create antibodies. Now, antigens can be complete or incomplete antigens. A complete antigen has two properties to it. One is immunogenicity. This means that your immune system recognizes it as foreign and it causes you to create more lymphocytes. It causes your immune system to react. And that's the second part, is reactivity. Lymphocytes and antibodies react to this foreign antigen. Okay, your immune system sees it, recognizes it as foreign, and you get rid of it. So that's immunogenicity and reactivity. So examples of complete antigens would be viruses, bacteria, fungi, even things like foreign proteins that get in your body, pollen. Your immune system recognizes that they're different. You generate more white blood cells, more lymphocytes to fight it off. Now we also have incomplete antigens, and another term for these is haptens. These are really tiny molecules, and by themselves, they're not immunogenic. So by themselves, they don't cause any problem. What causes a problem is when this tiny little molecule binds to a bigger molecule. Maybe in your body, maybe it's binding to something a little bit larger. And when that happens, that combination, your body will recognize as foreign. Now your body goes, oh, what is that? That doesn't belong, because now it's something that's big and foreign. And this causes your immune system to sort of react to it. And usually we call these hypersensitivities. So this would be anytime maybe you use a detergent and it causes your skin to break out in a rash, or maybe you've used a new lotion that causes that. Poison ivy oil. Your immune system is sort of overreacting to something that really is by itself not a problem, but what's happened is it has combined with proteins in your skin or combined with bigger molecules in your body, and now your body goes, hey, that doesn't belong. We got to fight that off. We've got to get rid of this. And it causes you to have this hypersensitivity. Now, when we talk about antigens, I also want to bring up this term up here at the top called an antigenic determinant. This is the part of the antigen, the foreign antigen, that's immunogenic. This means this is the part where antibodies and T cells are going to bind to it. Okay, so a lot of times what we'll find is that most antigens have lots of antigenic determinants. And so what that means is that if a foreign antigen gets in your body, you're going to create multiple B cells might recognize it. Multiple T cells might recognize it as foreign and fight it off. So a lot of times it'll mobilize several different lymphocytes. And this is also going to be why you might make several different antibodies to fight it off as well. Big proteins have hundreds of antigenic determinants. So you might have multiple B cells pumping out different kinds of antibodies and multiple T cells trying to fight it off. Tiny, chemically simple molecules, even large chemically simple molecules, plastics, surgical steel, these don't have any immunogenicity. So this is why a lot of times they'll use these to make implants, because your body's not going to recognize these as foreign. You're not going to try to fight these off. So this is a great example. This is showing you an antigen. So here's a foreign antigen, and you can see that we have three different antigenic determinants here. So here is one, here is one, and here is one. So this one foreign molecule right here has three different antigenic determinants, right? The shape is different. So what that means is that you might have three different B cells that activate to this and make three different antibodies. You have an antibody that's shaped like this that can bind to it. You have a completely different antibody shaped like this determinant that can bind, and another one shaped like this that can bind. So you have multiple antibodies now that can bind to this exact same foreign antigen. Okay? That's what antigenic determinant means. We also have self-antigens. So remember, a lot of time as we're walking through this chapter and we talk about antigen, we're referring to foreign antigens. They're foreign things that are getting in your body, but we do have self-antigens. These are the fingerprint. So this is the sugar coating on the surface of our cells. It's how your body knows what belongs. And your cells are either self or non-self, right? If it's a self-antigen, it means that your body recognizes it as self. So it's not antigenic to yourself, but it is antigenic to somebody else, because we all have very unique self-antigens. Mine are different from yours. And so if I needed a kidney transplant, even if we were a good match, we had the same blood type, some of our other proteins were similar, that's what good match means. If I got somebody else's kidney, it does not have my self-antigens. Unless it's coming from my identical twin, which I don't have, it is not going to contain my self-antigens on the cells of that organ. So my body has the potential to recognize it as foreign and fight it off. Okay, so we all have this very unique fingerprint, this self-antigen. And so an example of a self-antigen are what are called MHC glycoproteins. MHC stands for major histocompatibility complex. Again, these are unique to an individual. They are basically like the fingerprint on the surface of your cells. And so these have a group that can hold a self or a foreign antigen. These actually play a huge role in your immune system. And we're going to come back to these when we talk about T cells. So remember how I told you that B cells and T cells, even though they're both part of our adaptive defenses, B cells make antibodies that fight off free-floating foreign organisms. T cells fight off body cells that don't belong, body cells that have been affected. How does a T cell know what body cell doesn't belong? It knows because of MHC glycoproteins. So a T cell can only recognize an antigen if it's presented on this MHC glycoprotein. That's the only way it can recognize it. And it will either recognize it as self or foreign. Okay. So we're going to come back to that. But here's an image that's showing you this. So you can see at the top, here's our body cell in yellow. And you can see the MHC proteins on the surface. Okay. And so let's say this body cell gets infected. So here is this foreign microbe over here, and you can see it has foreign antigens all over the surface that don't belong. This body cell gets infected with that bacteria. Okay. It takes the foreign antigen and puts it on that MHC protein. That's how a T cell knows that that cell's been infected. Okay, so these MHC proteins are incredibly important in our immune response. That's the only way that a T cell would know that cell's been infected. And so when we talk about our adaptive immune system, there's really three kinds of cells. There's T cells, B cells, and then antigen-presenting cells. So let me go back. This is an antigen-presenting cell. So again, this is the only way a T cell knows that a cell in your body has been infected by a virus or a bacteria. So we say that antigen-presenting cells, they don't respond to very particular foreign antigens, but they do play a really essential role in activating your T cells. Okay, so that's why I drew that line up there. So we're going to walk through our B cells and our T cells, which remember, these are lymphocytes. These are a type of white blood cell. Remember, all of your white blood cells come from red bone marrow. In fact, all of your formed elements do. When a lymphocyte becomes mature, it has two things. One, it has immunocompetence. That means that it can recognize one specific foreign antigen and bind to it. Okay. Lymphocytes have only one unique type of antigen receptor. I'm going to underline that. They have one unique type of antigen receptor. This is where specificity comes in. That means that you have in your body billions of different kinds of lymphocytes, each one recognizing a different unique type of foreign antigen. This is why new emerging infectious diseases are so dangerous to humans, because we don't have lymphocytes that recognize new foreign antigens. We have lymphocytes that recognize ones that our ancestors have been infected with. That's how our body knows what to make. Okay, so new diseases can be detrimental to humans because lymphocytes are very specific. They bind to only one foreign antigen. So you may have a lymphocyte in your body that hangs around your whole lifetime and is never activated because you're never infected with that particular foreign antigen, okay? They also exhibit, when they are mature, self-tolerance. That means they do not respond to your own self-antigens. So we say that genes in your body, genes in your DNA, not antigens, determine which substances your immune system will recognize. So again, what kinds of lymphocytes you have in terms of what antigens they will recognize is determined genetically by what your parents and your grandparents and your great-grandparents and your great-great-great-grandparents have been infected with. Okay, that's how your body knows what kinds of lymphocytes to make out, to produce, what kinds of foreign antigens your body might see, okay? Only some antigens, I mentioned this, will ever invade our body. So you might have some immunocompetent cells that stay idle for a lifetime. They're never activated, they don't need to be. And again, we have over 1 billion different types of antigen receptors. So let's talk a little bit about our B cells. B cells are made in bone marrow. All of your formed elements are made in bone marrow. Again, B cells are going to respond to bacteria, bacterial toxins, viruses that are free floating. Again, these are floating around in your body's humors, in your lymph, in your blood, in your interstitial fluid. And the job of a B cell is it's going to pump out an antibody to fight off that free floating foreign organism. T cells are made in our bone marrow. All of our formed elements are made in bone marrow, but T cells mature in the thymus. So I remember the T in T cell for T in thymus. So once T cells are made in the bone marrow, they go to the thymus to become mature. And the process of maturing a T cell is incredibly complicated. And I want you to think about why that might be. Unlike B cells, which are just going after free-floating foreign organisms, they're pumping out antibodies to make and go get rid of, flag those free-floating foreign organisms for destruction. T cells are killing infected body cells. The last thing you want is a T cell that starts killing your own body cells that are healthy, right? So when T cells are made, they go to the thymus where they are basically put through multiple tests to ensure that they do not recognize self-antigens as foreign antigens. Because of that, only about 2% of the T cells that you make survive and make it out into circulation. The other 98% of the T cells that your body makes fail the test, and they end up recognizing your own body cells as foreign, and you take them out of circulation. They're never put in circulation. So again, remember, T cells are going after body cells that are infected with viruses or bacteria, or body cells that have changed via cancer, or transplanted cells. And they're going to attack that cell directly. So again, the last thing you want is a T cell that sees your own body cell that's healthy as something it needs to get rid of. Can it happen? Yeah. We call those autoimmune diseases. And that is where your T cells attack your own healthy body tissues. We don't see them a ton, but we see them, so it can happen. So this is a table from your book that's just kind of showing you the difference between B cells and T cells. So again, the type of response that we find with our B cells is humoral immunity, because your B cells make antibodies that float through your body's humors. Okay, so making antibodies. The targets are free-floating foreign organisms. So we say extracellular pathogens. So maybe viruses, bacteria, bacterial toxins. They're all going to be made in the bone marrow, but B cells also mature in bone marrow. So B for bone marrow. T cells provide us with cellular immunity. They do not make antibodies, right? And they are going after infected body cells. So we say intracellular pathogens. Intra means within. So these are going after pathogens that are already in your body's cells. Again, site of origin for all formed elements is bone marrow, but T cells mature in the thymus. T for thymus.
So when things go wrong in the body and the body can't handle it, it's got to have outside help, like, I don't know, a drug or something.
Yeah.
Is it the lymphocytes and the B sites going bad, or does it depend on what is attacking the body? So it could be a combination of both or one or the other? Or...
Yeah. So it is, maybe you have to have an antibiotic. Like maybe you have an active infection and you need an antibiotic to kill it off. Maybe you have a bacterial infection. The antibiotic is just going to work. It's just going to help your immune system. It doesn't mean that your B cells aren't working or your T cells aren't working. It's just going to heal the infection faster for you. Okay. And it's going to prevent tissue necrosis and all kinds of other things.
So I guess what I'm trying to understand is it just depends on what's attacking the body.
Yes.
That it makes it depend on what extra help you need.
Right. So, some bacteria are just like, if we're talking about bacteria, some bacteria are really good at evading the immune system. Some of them are just nasty. There are some bacteria that can actually wall themselves off and hide themselves, camouflage them from your immune system. So your immune system can't even see them and fight them off. So yeah, I think it just depends on what is infecting your body as to what kind of medication or what kind of help you might need. There are some viral infections that you get where there's really not anything we can do for you. It's really just like rest, let it run its course, and eventually your body will fight it off and get rid of it. But then there are other things where if you are infected, especially bacteria, where you probably will need antibiotics and some extra help and medication to fight it off. So I also told you that, even though we really think of our third line of defense, the adaptive immune system, as our B cells and our T cells, those antigen presenting cells. So let's get back here. These cells, anytime a body cell has been infected by a virus or bacteria, it has to be able to put that foreign antigen on its surface. When that happens, we say this now becomes an antigen presenting cell. This is essential for T cells. Since T cells, the only way they would know a body cell's been infected is if that body cell presents the foreign antigen to it and says, here, this is inside of me, this doesn't belong. And so I want to mention a little bit on these antigen presenting cells. Again, they're going to engulf the antigens. They're going to put the foreign antigen on the surface, on that MHC protein. And they'll find a T cell and tell the T cell, hey, this is what is inside some body cells. This doesn't belong. And it helps to activate those T cells. Because again, remember, T cells are going after infected body cells. So there's different kinds of antigen presenting cells. Dendritic cells like Langerhans cells. This is a type of cell, if you remember from Anatomy 1, that you find in your skin. Macrophages are types of antigen presenting cells. And this one's going to sound really weird, but even B cells can be antigen presenting cells. So let's walk through some of these. Dendritic cells. These will gobble up foreign organisms and then present that foreign antigen to a T cell. Again, these are going to be things like Langerhans cells. So we find them in the skin and the membranes. Once they gobble up something foreign, they'll get pulled into your lymphatic vessels and they'll find a lymphocyte. They'll find a T cell and tell that T cell, hey, this doesn't belong. This is in our body, and it activates those T cells. These are actually the most effective antigen presenter that we have in the body, are these dendritic cells. One of the things that makes them so effective, if you look at this image at the bottom, you can see that this cell has these long branching processes. That means it has a really long reach. So if something gets in, it is really hard to get around that kind of cell, those dendritic cells. We also have macrophages. We find these in lymphoid organs. We find them in connective tissue, which is all over our body. These will do the same thing. They will present that foreign antigen to the T cell, and it causes, macrophages are doing this for two reasons. They're kind of doing it for a selfish reason, because when a macrophage presents a foreign antigen to a T cell, it doesn't just activate the T cell. The T cell will release chemicals that make the macrophage become more voracious, and it starts eating more. And so they're kind of doing it for a selfish reason, because it activates them more and it puts them into hyperdrive. Okay, so it's activating not only your T cells, but it's making macrophages more voracious and gobble up more foreign organisms. And then B cells, like I mentioned, these can also act as antigen presenting cells. B cells and T cells really help to activate each other. And as we walk through B cells and T cells, you're going to see that. So that was sort of a 10,000-foot overview of your adaptive immune system. So again, it is B cells, T cells, which are lymphocytes, antigen presenting cells. These are going to help to destroy anything in your body that doesn't belong, any foreign antigen. And your adaptive immune system has to be able to recognize a foreign antigen and bind to it. And all those cells have to be able to communicate with each other so that they can help to increase whatever kind of response that needs to happen to get rid of the foreign antigen. So really what we're going to focus on today is the B cells. We're really going to spend most of our time today talking about the humoral immune response. So we're going to talk a lot about antibodies. We're going to talk about B cells. We're going to talk about vaccines and how those work, okay? So remember, B cells, these are part of our adaptive defenses. They're very specific, and they're targeting any free-floating foreign organism, extracellular targets. This is a great image. This is showing you up here. We have in red, this is a foreign antigen. So the first thing I want you to notice, it's free floating. It is not inside of a body cell. So this would be floating in your bloodstream, your lymphatic fluid, extracellular tissue fluid, any fluid in your body. Here, you have 3 different B cells. Only one of those B cells can recognize that foreign antigen. Only this one. This is the only one that has a receptor that is specific to that foreign antigen that got in the body. So remember, I told you that B cells and T cells have specificity. They only will recognize a very particular foreign antigen. This image is trying to show you that. Okay, so in this case, again, remember, you have billions of B cells and T cells. So this is showing you out of those billions, this one will recognize that foreign antigen. And that one will become activated. And once it's activated, once it binds to the foreign antigen, it will begin to proliferate, which that just means it will form clones. So basically starts making a whole bunch of itself. So now you can see we have more of that exact same type of B cell with that exact same receptor. Now, most of those clones will become a type of cell called a plasma cell. Plasma cells make antibodies, and they make a lot of them. They make antibodies at a rate of about 2,000 per second for 4 or 5 days. Okay, so let's go back to this picture. You see these clones. Most of these turn into this type of cell right here. Those are plasma cells. Plasma cells make antibodies. And I want you to look at those antibodies. They look pretty identical to the receptors that you find on that activated B cell, right? That's basically what they are. So these antibodies are proteins that have the same receptor, and these are going to latch on to that foreign free-floating antigen. That's their job. They're going to flag it for destruction. So these plasma cells are going to pump out antibodies at this huge rate of like 2,000 antibodies per second. Now it takes time though. It takes time. If we look at this picture, it takes time for this B cell to even recognize the foreign antigen. Then it's got to make clones. Then it's got to make plasma cells. Then it starts pumping out antibodies. All of that takes time. It takes days for that to happen. This is why when you get sick, you feel crummy for a couple of days. You are not going to feel good because your body is ramping up. It is trying to make those antibodies. So it takes time for that to happen, for those antibodies to start being produced. Once those antibodies start being produced, you will start feeling better. Once antibody levels get really high, you start feeling a lot better. But this is typically why it takes 7 to 10 days to get over a cold. Now, those antibodies are going to circulate in your body's humors, your blood, your lymphatic fluid, your tissue fluid, breast milk. Antibodies will circulate through all of that fluid, and they're going to bind to any free-floating foreign antigen and flag it for destruction. They basically inactivate it and flag it so that a macrophage or another cell can gobble it up. Now there are also, there's another type of cell that's created in this process. These are called memory cells. So I want you to look back at this picture. I told you that a lot of these cloned cells make plasma cells, which pump out antibodies. But notice we've also made another kind of cell over here. That's a memory cell. Memory cells provide you with exactly what it sounds like, immunological memory. So What this means is that if that exact same foreign antigen infects your body a second time, these memory cells are the ones that see it, and they will respond so much faster. They will make antibodies, and your antibody levels will be so high so quickly that you don't even know you were sick. You will have no symptoms from feeling the infection of that foreign antigen. And so this is what we call our primary and secondary immune response. Okay, so the primary immune response is what happens the first time you see a foreign antigen. So the very first time you see it, there is a lag period. It usually takes about 3 to 6 days before you really start making some antibodies. And that's just the time it takes for the B cell to recognize the foreign antigen, make clones, make plasma cells. It just takes a while. Peak levels of antibodies are reached at about 10 days. And then antibody levels will start to drop off. So here's an image of that. So we can see our primary immune response. I'll draw a line. We're looking at everything to the left of that red line. So you can see down at the very bottom, time 0, you are exposed to a foreign antigen. And you can see that it is taking, before antibody levels start to rise, it's taking about 5 days. And then your antibody levels will rise, or starting around days 5 or 6, you'll start to pump out antibodies. Antibody levels rise, you feel better, the infection is gone, antibody levels drop off within about 25 days. And that's our primary response. But remember, in our primary response, we have made clones, we've made plasma cells, we've made antibodies, but we also made memory. So the secondary response is if you are re-exposed to the exact same foreign antigen, this is where those memory cells come in. They respond within hours. Antibody levels will peak in 2 or 3 days, and they peak at much higher levels than they did during the primary response. The antibodies that are created from memory cells, these bind with greater affinity. And antibody levels during the secondary response will stay high for weeks to months. And these memory cells that are made during that primary immune response, they can hang around in your body for 20 years or more. And so this is how flu shots, this is how childhood vaccinations work. So let's look at this picture again. And now we are looking to the right of that red line that I just drew. Okay, so remember, everything in blue, this was our primary response. First time we saw the foreign antigen. Took a while to make those antibodies. You can see here, second time you are exposed to the exact same antigen, your antibody levels start climbing immediately. And notice how much higher they get. So you pump out way more antibodies during a secondary response than you ever did during a primary response. And again, this is why you don't even feel sick. You don't even know that you were infected. And so that's what this picture is showing you. So I'll draw a line right here. Everything above this line is our primary response. And everything below that red line is the secondary response. So you can see that same foreign antigen gets in, memory cells recognize it, and they pump out much higher levels of plasma cells and antibodies than you did the first time around. And again, those memory cells can hang around for a long time. So when we talk about the immune system, when we talk about our B cells, we have what's called active immunity and passive immunity. Active immunity literally means you are activating your immune system. So this is when a B cell encounters a foreign antigen and produces antibodies. So there's actually 2 kinds of active immunity. There's naturally acquired and artificially acquired. So naturally acquired is you get sick. You are infected with a bacterial or a virus. You get sick, you go through the primary response, you make memory, right? The other one is artificially acquired. Artificially acquired immunity still activates your immune system, But this would be an example of a vaccine. When you get a vaccine, you are getting a live, dead, or attenuated virus. So it's a virus that is either completely dead, and all it is a dead viral capsid that your body, they inject it into you, floats around in your humors. It's dead. It can never infect a body cell, but boy, it's free floating, and B cells can see it, and they see it as a foreign antigen, and they bind to it. They don't know it's dead, and they clone, and they make plasma cells, and they make antibodies, and they make memory cells. You have activated your immune system with zero threat because that virus was dead. It's never going to infect a body cell. Now, can you feel crappy after you get a shot, a vaccine? Yeah, because the whole process of primary immunity, ramping up your B cells, making memory cells, pumping out antibodies, it's going to make you feel crappy for about 24 hours. Okay, it's usually a lot less severe than if you actually got an active infection. Okay. So this is how vaccines work. Most vaccines, now this does not include mRNA vaccines, but most vaccines are dead or attenuated, which means weakened pathogens, which means they can't infect your body cells. And again, they're going to spare us the symptoms of a primary response. Yeah, you might feel crappy for 24 hours, but you're not going to feel bad for 10 to 15 days, right? But you've made memory. So the second time your body sees that exact same foreign antigen, you fight it off and you don't even know that you were sick. So this is how we've essentially eradicated things like smallpox, whooping cough, polio, measles, mumps, rubella, all of those things we really don't see much of anymore. About 65,000 Americans, and that number is probably higher every year, that number gets higher. They die each year from vaccine preventable infections. I'm probably going to add to this next week. I'll probably add a little bit about mRNA vaccines so that we can talk a little bit about those as well. Those are relatively new, obviously, with the COVID vaccine. So we'll probably add a little bit to this lecture for next Tuesday. Now, other than active immunity, we also have passive immunity. Passive immunity is basically when antibodies are just put in your body. You're not activating your immune system. Your B cells aren't recognizing a foreign antigen. They're not making clones. They're not making plasma cells. They're not making memory cells. All you're doing is injecting antibodies into your body to help fight an infection. B cells are never activated. Memory does not occur. This is passive immunity. Okay? The protection that you get will end as soon as those antibodies degrade. Antibodies are proteins. They usually will degrade within a week or two. Okay?
Is that like the monoclonal treatments that they were offering or are offering? Is that the same thing?
Yeah, so monoclonal antibodies, yeah. So when COVID first started, they were using people who had gotten COVID and then recovered from COVID. They were drawing their blood and giving very sick patients injections of those people's blood plasma. The blood plasma contained their antibodies to COVID that they produced. That's what we did immediately. Well, now they've been able to take those same antibodies and create them in a lab, and we call those monoclonal antibodies. So now instead of using somebody else's blood plasma, which we talked about blood, there's going to also be the risk when you use someone's blood plasma of any kind of bloodborne pathogen being passed. And so by having monoclonal antibodies, it's the same thing. You're getting the antibodies that they've been generated in a lab. So yes, that's passive immunity. So there's two types of passive immunity. There's artificially acquired and naturally acquired. So what we were just talking about, that's artificially acquired. It's injecting a serum of antibodies into somebody, like monoclonal antibodies. And again, the protection is immediate. You'll probably feel better immediately. So this is something where monoclonal antibodies right now, especially for COVID, are in short supply. So right now they are giving those to patients who are admitted to a hospital, patients who really need those to feel better. Okay. Naturally acquired passive immunity is what a mom who is breastfeeding her child might be providing. Not giving that baby an injection of anything, but remember, antibodies provide us with what's called humoral immunity. Antibodies flow through all of your body's fluid. So blood, lymphatic fluid, tissue fluid, breast milk. So when you breastfeed a baby, any antibodies that you have will be in that breast milk, and you will pass that on to your baby. That baby will then have passive immunity. And so when I had my son, I told my husband, I had him in March, and I was like, I'm gonna nurse him until I have to go back to work in August. And so I did. I nursed him until I went to work in August, and then he went to daycare. And I was like, oh gosh, I'm gonna nurse him while he's at daycare because like there's so many germs and there's just so much. And in the fall with flu season, it was terrible. So I nursed him through that. And then we got to December, and that was the year where the swine flu was really bad. So I was like, well, I just keep on going. And I ended up, I feel like I nursed him for 5 years. I didn't. It was 15 months, but it was really long. But if I did it because every time I thought, okay, we're gonna be done, there was something else that made me think maybe he needs that passive immunity. Maybe I should keep going so he has those antibodies. So again, there's active and passive humoral immunity. Active means you are activating your memory cells. You are activating your B cells. Your body is making antibodies. You produce memory cells so that you have protection the next time you see it. Naturally acquired means you just got sick. Or artificially acquired means you got a vaccine. Okay? Passive immunity. Passive immunity means that you're not activating your immune system. You're not making memory to anything. And this would be either getting an injection of antibodies or passing antibodies to a baby through breast milk. So I want to talk a little bit about antibodies in general. I know we've talked about how they float through your body's humors. Antibodies are proteins, and they're going to bind to foreign antigens. Again, they're made by our plasma cells, those B cells. We can also call antibodies immunoglobulins. I will call them antibodies. And these are capable of binding with a very particular foreign antigen that is free floating. Okay, so remember, they are specific. Now, because they're specific, your body has tons of billions of different B cells that make very particular antibodies, each one. So that means if you have billions of different B cells, you have the potential to make billions of different antibodies. All of those antibodies, we class them into 5 different classes called IG classes. Here are the classes. I always remember MAG. So it's IgM, IgA, IgD, IgG, and IgE. Okay, now you don't have to know names of particular antibodies or anything really about each of these classes. I just want you to know that of all the antibodies that our body can pump out of the billions, we classify them different ways. We classify them based on essentially what their receptor regions look like, and also what kind of foreign antigen they are targeting. The one that I do want you to know is number 5 up here, IgE antibodies. These actually play a huge role in allergies. So we'll talk more about these towards the end of the lecture. This is trying to show you an antibody. So I'm actually going to skip back to this picture back here. Notice when you see these antibodies, these little blue things, they're always shaped like little Ys, okay? And so in this picture here, we're looking at an antibody. It's shaped like a Y. We're just looking at it up close. And what I really want you to pay attention to is the ends of the Y. The ends of the Y have that darker color there. That is the antigen binding site. So that's the area where the antibody will bind to the foreign antigen. We also say that is the variable region. So depending on the antibody, that's the area of the antibody that will change depending on what foreign antigen it's being created for. So remember, antibodies do not destroy a foreign antigen all by themselves. They get released by plasma cells. Antibodies float through your body's fluids. And their job is they will latch on, they will grab on to a free-floating foreign organism. They help to inactivate it and they flag it for destruction, but they do not destroy it themselves. When an antibody latches on to a foreign antigen, they form something called an antigen-antibody complex. From there, how that foreign antigen will be destroyed is one of the 4 ways I have listed here. Okay, so once it's flagged for destruction, once an antibody binds to it, it can be killed by what's called neutralization. It can be killed by agglutination, precipitation, or complement fixation. So I'm going to walk you through what each of those means. So the first one, if antibodies bind to this free-floating foreign antigen, flag it for destruction, they're going to maybe bind to a virus, free-floating virus, bacteria, bacterial toxin. But typically what they do is when they bind to it, they neutralize it so it's no longer harmful. So here in this picture, I circled it in red. You can see that we've got a foreign antigen up here. We've got some antibodies here. When the antibodies bind to that foreign antigen, so in this case, this might be like bacterial toxin. When they bind to that toxin, what they're doing is they're coating all around the toxin. By coating all around the toxin, they're neutralizing it, so it is no longer harmful to your cells and tissues. And then from there, it'll go through phagocytosis. You'll have macrophages and other phagocytic cells that now know that doesn't belong, it's been flagged, let's gobble it up. Okay, but in the meantime, antibodies have neutralized it. It's not harmful anymore. They can also go through a process called agglutination. You know this one. This is where antibodies will bind to foreign antigens on cells and cause them to clump. So a great example of agglutination is the clumping of mismatched red blood cells. So here in this picture, you can see that we've got all of these antibodies binding to the foreign antigen on the surface of these red blood cells. And it's causing a whole bunch of red blood cells to clump up together. That's called agglutination. Those cells are flagged for destruction and phagocytic cells will gobble them up. So remember when we talked about mismatched blood transfusions, I told you that when someone gets a blood transfusion of blood that does not match, a lot of times what happens is the red blood cells, it causes a lot of clumping. The clumping can clog vessels in your body, so you're not able to supply blood where it needs to go. A lot of those mismatched red blood cells will burst open, get phagocytized, and you end up with a ton of extra hemoglobin in your body, which can shut your kidneys down. Okay. Another one is precipitation. So this is going to happen when you have big soluble molecules. A soluble molecule is a molecule that's dissolvable in solution. And so you get the soluble molecule where antibodies binds to them and it causes them to settle out of solution. I want you to think of chemistry. If you ever took a chemistry class, precipitation. When something precipitates out of solution, it means that you take something that's soluble, that's dissolvable, and you precipitate it out of solution. It turns into something that can fall out of that solution. And that's exactly what's happening. So you can see here in that one that's circled red, little soluble antigens, and you can see all those antibodies binding to it, making it bigger and making it precipitate out of solution. Again, phagocytic cells will gobble it up. So notice in every single one of these, what is in common is the fact that the antibodies aren't killing off that foreign organism. Instead, they're flagging it for destruction. Phagocytic cells get rid of it. Okay? And then the last one is complement fixation. This is the main antibody defense against a cellular antigen. So a free-floating bacteria, a mismatched red blood cell, free-floating, well, a virus isn't cellular, but this is complement fixation. This is how you're going to kill off a free-floating living organism that gets in your body that doesn't belong. Okay? And so remember when we talked about our second line of defense, we talked about complement proteins and how we have like 20 different complement proteins that circulate in our blood. 5 of those proteins can come together and create something called a membrane attack complex. That's what this is. So typically what will happen is the antibodies will bind and they're going to trigger that membrane attack complex to come and punch a hole in that flagged cell. So here's an image. Okay, so you've got a cell, so maybe a bacterial cell, maybe a red blood cell, and you've got antibodies binding to the foreign antigen on the surface. Okay? This is saying, hey, this is free floating, this is in our body, this does not belong, I'm flagging this for destruction. Here comes from our second line of defense, that membrane attack complex. We get 20 of these proteins, these complement proteins, 5 of those will come together, create that membrane attack complex and punch a hole through that cell. That's going to cause water to rush into the cell and the cell will burst open. So again, remember, when we think about incompatible blood transfusions, when you get a blood transfusion, if it doesn't match your own blood type, one of the problems is agglutination. So antibodies are going to cause it all to clump and clog small vessels. The other thing that's going to happen is antibodies are going to flag those red blood cells that don't belong. Complement proteins will punch a hole. That red blood cell will burst open, release all of that hemoglobin, and that will shut your kidneys down. Okay, so 2 things happen there. Now we mentioned this already. This is the monoclonal antibodies. So remember, this is basically where anytime they can produce a pure antibody commercially. And I want to talk a little bit about what these are, how companies produce monoclonal antibodies. So they're actually produced by something called a hybridoma. These are cell hybrids. So basically what they do is they fuse a tumor cell. I want you to think about tumor cells, think of cancer cells. Cancer cells have uncontrolled cell division. They go through mitosis uncontrollably. They fuse a cell that replicates uncontrollably and a B cell. And they put the 2 together. Now you have a B cell that's going to replicate uncontrollably and proliferate indefinitely and have the ability to produce a single type of antibody. This is how we get monoclonal antibodies in a lab. So again, a lot of times these are used for research. These can be used as treatment for people who are ill, especially with viral infections. Can even be used as cancer treatment. So monoclonal antibodies are incredibly important type of treatment. So in summary, just in terms of our antibodies, so the antigen and antibody complex. So when an antibody binds to an antigen, remember it's not destroying the antigen, it's flagging it for destruction by one of these 4 ways listed here. Okay, it flags it for neutralization to get rid of it through phagocytosis. Those antibodies cause cells to agglutinate. Phagocytic cells gobble them up. It flags it and it will pull out soluble molecules. Phagocytic cells will then gobble them up. Or it flags it for complement proteins to punch a hole and cause the cell to lyse. So keep in mind that antibodies are incredibly important, but they don't destroy the foreign antigen all by themselves. They need help to do that. They need phagocytic cells to help them with that process. They need complement proteins to help with that process. Again, remember, antibodies are only able to see a free-floating foreign antigen. Antibodies are not going to be able to see a foreign antigen that's inside a body cell. They can't see that. Okay, so if a body cell has been infected by a virus, antibodies can't see that. They can only see it when it's free-floating. Free-floating in tissue fluid, blood, or lymph. Now, typically, antibodies cannot invade solid tissue unless there is a lesion present that allows the antibody to get in. Okay.